Okay, so let's try and unpack this. There's this timeline we all kind of have in our heads when it comes to alcohol and the human body. It's pretty binary, right?
Right.
You've got the immediate timeline, the fun part, the buzz, being a social lubricant, and then inevitably the headache the next morning, the classic hangover.
Right. The acute effects. We know that chapter very, very well.
Exactly. And then way, way down the road, you have the scary timeline. We're talking 30, maybe 40 years of heavy use.
Yeah.
Liver failure, cirrhosis, total system collapse. We know the beginning and we know the catastrophic end.
Those are the two chapters everyone reads. The
Mhm.
morning after and then decades later.
But this is where it gets really interesting. There's this massive black box in the middle. I mean, huge. What is happening in that intermediate phase? What is your body actually doing after, say, just 6 months of daily drinking? Is it just a slow, linear slide toward damage? Or is the body doing something stranger?
It's a fantastic question. And honestly, until pretty recently, it was a surprisingly difficult one to answer. We just assumed it was, damage accumulation like rust slowly eating a car. But the reality is, it's much more complex.
Well, we are cracking that black box open today. We've got a fascinating study published in the FASEB journal that shines a light right into that gap. The title is a bit of a mouthful.
Oh, it is?
Short-term alcohol consumption is sufficient to disrupt microbial communities and the intestinal and circulating metabolites in a non-human primate model.
It doesn't exactly roll off the tongue, does it?
It does not.
Yep.
But the mission of this deep dive is to explore what the researchers call the hidden organ, the gut microbiome. And the headline here is that a relatively short period of drinking, just 6 months, doesn't just damage the gut, it completely rewires the body's chemical signaling.
From the intestine all the way to the blood,
Yeah.
maybe even the brain.
And what makes this particular deep dive so valuable is the source material. We aren't looking at surveys where people essentially guess, or let's be honest, lie about how much they drank last week.
Right. And we aren't looking at mice.
That's a crucial distinction. Let's start there. The setup. This study used rhesus macaques. Now, I can hear the listener asking, why do I care about monkeys? I'm not a monkey. Why was this specific choice so important?
It all comes down to, translation. How well do these results actually apply to you and me? Human studies on alcohol are they're notoriously messy.
Mm-hmm.
You can't control what people eat. You can't control their stress levels. And like you said, self-reporting is just not reliable. Plus, you can't exactly take deep tissue biopsies from a healthy person's intestine just for fun.
Right. Pass the potatoes, and oh, do you mind if I snip a piece of your colon?
Yeah,
Not a great dinner party conversation.
precisely. So science usually turns to mice. They're the standard lab model. But mice are biologically very different from us. Their gut microbiomes, for one, are not structured like ours. And here's the kicker. Mice naturally avoid alcohol.
They do. I thought they'd eat anything.
They generally hate it. So to get a mouse to reach the same blood alcohol concentration as a human, you often have to force it. You have to give them ethanol dosages 2 to 5 times higher than what a human would voluntarily drink.
Wow. So you're basically pickling the mouse just to get the data point.
In a way, yeah.
Hmm.
And that creates a level of toxicity and stress that might not reflect what actually happens when a human voluntarily, sits down for a few beers.
Okay.
But rhesus macaques, they share significant genetic and physiological traits with us. Their gut microbiomes are remarkably similar to ours. And crucially for this study, they have a natural propensity to consume alcohol.
They like a drink.
They do, which allowed the researchers to set up a voluntary self-administration model. They didn't force-feed the animal.
So they just put it out for them.
Pretty much. They gave the macaques access to a 4% ethanol solution. Think of it as the strength of a light beer and fresh water for 22 hours a day.
So they had a choice. They could drink the water or they could drink the beer.
They had a choice. And they did this for 6 months. Now, in the lifespan of a macaque, and in the context of this research, that's defined as short term. It mimics that middle period we were just talking about.
Okay, so we've got the perfect model. Controlled environment, voluntary drinking, light beer strength, 6 months. Now let's get into the weeds, literally. The hidden organ. We always hear about the liver taking the hit, but this study was obsessed with the gut. What actually happened to the ecosystem down there?
The first major finding is a shift in what's called beta diversity.
Beta diversity. That sounds like a metric from a corporate HR seminar. We need to improve our beta diversity. What does that mean in a biological context?
It's a good way to put it. Think of it as the landscape view. If you walked into a forest, alpha diversity would be like counting how many different species of trees there are. Beta diversity is looking at the entire ecosystem structure, the trees, the shrubs, the undergrowth, and seeing how that whole landscape compares to a different forest.
So after 6 months of drinking, the forest of the gut looked completely different.
It was a different world. The alcohol caused a structural reorganization of the entire bacterial community. The membership changed, the dominance of certain groups.
It all shifted.
Now, usually when we hear about alcohol and bacteria, the assumption is simple. Alcohol is a disinfectant. We use it to clean wounds.
Yeah.
So logically, it kills the good bugs and the bad bugs take over.
Yeah.
Is that what happened here?
See, that's the logical assumption. But biology is rarely that simple. It's much more nuanced.
Okay.
There was definitely a decrease in some beneficial bacteria. Specifically, the researchers saw a significant reduction in bacteria that produce short-chain fatty acids, or SCFAs. We're talking about like Eubacterium and Subdologranulum.
And just to clarify for everyone, SCFAs are the good guys.
Right. Essential. They are the primary fuel source for the cells lining your colon. They keep the gut wall strong and tight. If you lose them, your gut becomes permeable. People call leaky gut. So seeing those bacteria drop is usually a massive red flag. Okay, so far, it sounds like the damaged narrative we expected. But then, I was reading the notes, and there seems to be a paradox.
Yes, the lactobacillus paradox. Now, you see lactobacillus on every yogurt container in the grocery store. It's the poster child for good bacteria.
Right, it's a probiotic. We pay extra for it.
Well, in this study, while the genus as a whole shifted, the researchers found that the abundance of specific lactobacillus strains was actually positively correlated with the amount of alcohol consumed.
Wait, let me get this straight. The more they drank, the more of this specific good bacteria appeared.
Exactly. It was dose dependent.
Yeah.
More alcohol equaled more lactobacillus.
That doesn't make any sense. Why would alcohol, which is stressing the system, encourage a probiotic?
This is where we have to stop thinking of the body as just a passive victim and start seeing it as a survivor. Lactobacillus has a very specific function. It stimulates mucus secretion. It fortifies the physical barrier of the intestine. So the expert consensus here is that this is a compensatory mechanism.
A compensatory mechanism. So the body's fighting back.
Exactly. The body realizes the gut wall is under attack from the ethanol. So it doesn't just sit there. It actively recruits or creates an environment for lactobacillus to thrive, essentially saying, we need more mucus, we need to thicken the walls, reinforce the castle.
It's like the body is scrambling to put up sandbags against a flood.
That's a perfect analogy. And that theory, the sandbag theory, it holds up when we look at the other bacteria that increased. We saw a rise in Prevotalaceae, Faecalibacterium, and Senegalomycelia.
Those are some serious Scrabble words, but what's the common thread between them?
The common theme is inflammation regulation. Prevotalaceae is known to inhibit inflammatory processes. Faecalibacterium is often found to be low in people with inflammatory bowel disease. So having more of it suggests the body is trying to create an anti-inflammatory state. And Senegalomycelia has been linked to regulating blood pressure.
Regulating blood pressure. But alcohol raises blood pressure.
It does. So seeing a bacteria rise that helps regulate it, again, it looks like the body's trying to balance the scales.
This is a huge aha moment for me, because I think most people assume that from the very first drink, your body is just inflamed and dying. But you're saying that in this short-term 6-month window, the microbiome actually shifted toward an anti-inflammatory state.
Precisely. The microbiome didn't just collapse, it re-engineered itself. It shifted its population to try and handle the stress. It's an adaptive response.
That is wild. It's like the body is in damage control mode, actively recruiting the peacekeepers.
But the story doesn't end with the bacteria. They're just the factories. We have to look at what they're producing. We need to look at the metabolites.
Right, section 3, the metabolites. I like to think of these as the chemical text messages the bacteria send to the rest of the body.
That's a great way to put it. They're the small molecules left behind after metabolism. The researchers used mass spectrometry to basically weigh the molecules in the feces to identify them. They were reading the logs of these messages.
And what were the texts saying? Help me, we're drowning.
Surprisingly, no. The texts were saying, stay calm. They found a significant drop in medium and long-chain fatty acids, specifically things like palmitic acid.
Palmitic acid. I feel like I've heard of that in nutrition circles, usually in a bad context.
Usually, yes. High levels are linked to liver disease, rigid cell membranes. It's very pro-inflammatory. So finding a decrease in palmitic acid
Means less inflammation.
Exactly. It aligns perfectly with the bacteria we just talked about. The signal being sent is reduce inflammation. Then they looked at amino acids and found increases in products like urocanic acid.
Urocanic acid. Sounds like something that would burn you.
Actually, it's like a sunscreen for your immune system. Urocanic acid has properties that interfere with the inflammasome.
The inflammasome. Okay, that definitely sounds like a bad superhero movie villain. Beware the inflammasome.
It's basically the body's internal fire alarm. It triggers swelling, heat, attack responses. Urocanic acid helps mute that alarm. So again, the gut is pumping out signals to suppress the immune response.
Okay, so we have a clear picture of the gut. The bacteria are shifting to protect the wall with mucus. They're producing chemicals that tell the local area to chill out. But the gut isn't Vegas. What happens there doesn't stay there.
No, it certainly does not. The gut is the portal to the blood.
So did these signals go systemic?
That is the million-dollar question. And to find out, the researchers analyzed the plasma, the blood, and yes, it was a full-body broadcast, but with some specific markers in the blood that are frankly mind-blowing. They connect the gut to organs you just wouldn't expect.
Let's talk about beta-hydroxybutyrate. This one stood out to me in the notes because my brother won't stop talking about his keto diet, and he loves this word. It's a ketone body, right?
It is. Beta-hydroxybutyrate increased significantly in the alcohol-consuming macaques. Now, we usually think of ketones as just alternative fuel for when you aren't eating carbs. But this molecule does something else. It acts as an inhibitor for histone deacetylases.
Okay, hold on. You're going to have to unpack that. Histone deacetylases. That sounds incredibly technical. Why should the listener care about that specific mechanism while they're driving to work right now?
Because it means epigenetics.
Yeah.
It means changing how your genes are read. Think of your DNA as a massive library of blueprints. Histone deacetylases are like the librarians deciding which blueprints get locked away and which ones get used.
Okay.
When you inhibit them, you are essentially flipping switches on your DNA. And specifically, this mechanism has been linked to changes in the brain in the amygdala.
The amygdala, the fear and anxiety center.
Yes. This connects the gut directly to the brain's emotional regulation.
Walk me through that path one more time.
Okay. You drink alcohol. The gut and liver process it. This produces beta-hydroxybutyrate. That molecule travels through the blood, it crosses into the brain, and it potentially flips genetic switches in the amygdala that regulate anxiety and behavior.
That is terrifying and amazing. So drinking isn't just relaxing you because you're buzzed. It's chemically rewriting the anxiety centers of your brain via the gut.
It suggests a physical pathway for that, yes. It changes the hardware, not just the software.
Yeah.
And it wasn't the only signal. They also found increased urate.
Urate. Isn't that what causes gout?
It is. High uric acid leads to gout, and it can be inflammatory in the lungs. But, and here's the duality of biology again, urate is also a potent antioxidant. It's neuroprotective. In the brain, it might actually be shielding neurons from damage.
So we have a signal that might cause gout in your toe, but might protect your brain cells.
It shows how hard the body is working to balance this equation. It's a trade-off. I'll risk the gout to save the neurons. We also saw increases in acetylcholine and kynurenate, both of which modulate neurological pathways.
So if we synthesize this, we have gut bacteria shifting to peacekeepers, fecal chemicals saying don't inflame, and blood markers that are tweaking brain genetics and balancing protection with toxicity.
And the plasma also showed signs of increased fatty acid oxidation and purine metabolism. Simply put, the metabolic engine is revving high. The body is working overtime.
This leads us to the big conflict, Section 5. I feel like we've been painting a surprisingly rosy picture of alcohol here. We're using words like anti-inflammatory and neuroprotective. If I didn't know better, I'd think the study was funded by a brewery. But we know alcohol is bad. Why is this data showing the opposite?
This is the critical distinction. And if listeners take one thing away from this deep dive, it should be this: the difference between short-term and long-term.
So 6 months is still the honeymoon phase for the body.
Biologically, yes. The researchers emphasize that chronic, long-term abuse, years and decades, leads to a hyper-inflammatory state. That's when the gut leaks, cytokines flood the system, organs fail. That's the end stage.
But this study
at 6 months, revealed a hypo-inflammatory phenotype. The immune system, specifically the white blood cells called monocytes, had a dampened response.
Dampened. So it's not that there isn't damage happening, it's that the body is actively suppressing the reaction to it.
Exactly. The body is smart. If you have a chronic low-level irritant, you don't want your immune system constantly exploding. That would kill you faster. So the body dampens the signal. It creates a shield.
It's a bunker mentality.
Correct. The microbiome changes we talked about,
Right.
more Prevotella, less palmitic acid, they align with this dampened state. The implication is that the body has this distinct adaptive phase. When you start drinking regularly, it doesn't just roll over. It tries to compensate.
But shields are heavy, and bunkers aren't exactly healthy places to live.
No, they are not.
Oh.
Maintaining this hypo-inflammatory state is metabolically expensive. It requires a total reorganization of your internal systems. And here's the danger. While your immune system is dampened to handle the alcohol, it might be less effective at fighting off other things.
Right. And eventually the bunker collapses.
The prevailing theory is that the damage we all associate with alcoholism comes when this compensatory system finally breaks down. The shield shatters.
And then the floodwaters come in. But what this study shows, and this is the scary part, is that the reprogramming happens fast. It doesn't take years. Within 6 months, these macaques were completely rewired.
It really challenges the way we think about tolerance, doesn't it? We think of tolerance as, okay, drink 3 beers now instead of 2. But this suggests a biological tolerance.
Yeah.
Your entire metabolism bending over backwards to accommodate the poison.
It makes me think about the functioning alcoholic stereotype, the person who drinks every day but seems fine, holds down a job, looks healthy.
On the surface, yes. But inside, this study suggests their microbiome is working overtime to maintain that facade. They're in that hypo-inflammatory defensive crouch.
Until they aren't.
Until the system is overwhelmed.
So let's wrap this up. We've gone from macaques drinking their daily 4% ethanol to a total restructuring of the hidden organ. We've seen the rise of peacekeeper bacteria, the chemical texts telling the body to stay calm, and epigenetic signals reaching the brain.
It's a powerful reminder that alcohol affects the body as a whole system. It's not just a liver toxin. It's a systemic modulator that triggers this highly organized response.
And the relevance to you, the listener. This happens fast. You don't have to be a lifelong drinker to have rewired your gut. 6 months was enough.
Absolutely. The hidden organ is sensitive, and it reacts quickly. The adaptation is impressive, but it's a warning sign, not a permission slip.
I want to leave everyone with a final thought to mull over. We talked about this shield, this anti-inflammatory state the body creates to survive short-term drinking.
The hypo-inflammatory phenotype.
Right. If short-term drinking creates this shield that eventually breaks into chronic disease, at what specific point does that shield shatter? Is it year 1, year 5? Does it depend on your genes?
We just don't know the exact tipping point.
And here is the really unsettling part. Are those calm down signals in your blood actually masking the damage until it's too late? Is the silence of your immune system a sign of health? Or is it just the calm before the storm?
That is the question. And relying on that silence might be the most dangerous gamble of all.
Something to think about next time you're debating that second drink. Thanks for listening to The Deep Dive. We'll catch you on the next one.
Stay curious.